Swayam Prabha. Digital India. Educated India.
Hi everyone, welcome to the next class of structural biology, and today we are going to talk about the next part of structural biology techniques where we have ended in the last class. So, as I told about the techniques of structural biology, X-ray, NMR, electron microscopy and low resolution techniques like mass spectrometry, co-purification, proteomics, CD and all. Today we are continuing where we ended the discussion about the technique X-ray crystallography. The goal of X-ray crystallography is viewing the images of biological macromolecules. So, what is the requirement? In order for an object to be visible under magnification, the wavelength lambda of the light must be, roughly speaking, not larger than the object. Okay. So, what is the problem? The problem is visible light, which is 400 to 700 nanometer, cannot produce an image of protein molecule which is bonded atoms are about 1.5 angstrom apart. If you think a protein molecule is made of carbon, nitrogen, oxygen and bonded with sulfur and phosphorus. So, most of the bonds are ranging from 1.5 angstrom around, and it is impossible for light microscopy to go there and help you visualize that. So, you need other things. Electromagnetic radiation is the other thing. Electromagnetic radiation having wavelength falls into the X-ray range. So, X-ray, why X-ray? Because it is another form of electromagnetic radiation, and if you look at the electromagnetic spectrum from radio frequency to microwave to infrared to visible light to ultraviolet to X-ray, you will see that the energy from radio to X-ray, it is going from low to high. The frequency in correlating to energy also going from low to high from radio to X-ray. The wavelength is inversely proportional, so it is going from smaller short wavelength to long wavelength. And if you look at the visible light, it is 400 nanometer, and for X-ray it is around 1, 2, 3 angstroms. So, electrons, as in electron microscope, are not form of electromagnetic radiation, but they still have wave like character which is needed for this. And in de Broglie, if you see, the wavelength is 0.1 angstrom. Unlike photons, electrons are charged, so they fry the specimen faster. So, the two things you are going to look here, the energy for X-ray is high and X-ray could damage the protein. So, these are two negative points, disadvantages of this technique which we are going to discuss later. But now, coming to the resolving power, it is increasingly enhancing, coming to radio frequency to microwave to infrared to visible to ultraviolet to X-ray, and in X-ray, as we talked about, we could see in the atomic resolution. So, why cannot we visualize molecule directly using X-ray? There are people who always want to go for straightforward. So, they want to ask, okay, we understand that lower wavelength is required, we understand that the radio frequency is required, it should be looked at the atomic resolution, which is not possible for light microscope. So, why not have a X-ray microscope? That is because a single molecule is very weak scatterer of X-ray. If you look at the technique, we will go beyond, we will go into in-depth analysis. You will see that the basis of X-ray crystallography technique is the X-ray is coming, it is hitting carbon, nitrogen, oxygen in the crystal, and it is diffracting. The diffraction or the scattering is the one which is giving us information. when you consider direct viewing, you are looking at one molecule, a single molecule, and a single molecule is a very weak scatter of X-ray, and most of the X-rays will pass through the molecule without being diffracted. So, the diffracted rays would be too weak to be detected. Is there any solution? Analyzing diffractions from crystal instead of single molecule. That is the reason we are going from understanding a small molecule, a single molecule of protein to a crystal. We need a crystal because a crystal have a lot of molecules. A crystal is made of three-dimensional repeat of ordered molecules 10 to the power 14 approximately, whose signal reinforce each other. The resulting diffracted rays are strong enough to be detected. So, if you compare now, if you look at, forget about X-ray, compare about one molecule and 10 to the power 14 molecule, you could easily get your answer. The 10 to the power 14 molecule is much better scatterer. So, the signal would be enhancing into a very significant amount from a single molecule, and that is where the success of X-ray crystallography is hidden. Unlike visible light, X-ray cannot be focused by lens also. The refractive index of X-ray in all materials is very close to 1. So, you cannot make a difference. How we could make a difference here, how we could make a solution of the problem? We use computer to simulate an image reconstructing lens. So, instead of putting a lens in case of light microscope or electron microscope, we are using computer, and the computer simulation through Fourier transformation is used as a lens here. The computer plays the part of the objective lens, computing the image of the object, then displaying it on the screen. So, you put your data and computer simulate it, and by simulating they are processing it like the lens is processing the light or the electromagnetic wave in light microscope or in electron microscope. So, what are the nature of the crystals? Under certain circumstances, biological macromolecules like protein, DNA, RNA can form crystal. It is not each and every condition or common condition. We will discuss this. Forming crystal is the main factor to get the technology to be forwarded, because in many cases, actually in most of the cases, as statistics says, only in 8% cases when you are doing everything correct, after that only 8% cases you are finding lucky to get a crystal. The resulting crystal is a three-dimensional array. As I told, crystallization means you have small molecules, but they are dispersed in the solution, but in crystal they come into a certain array and a three-dimensional array. You could look this as a natural crystal. You could see that crystal of small molecule which are actually salts. This is a ice crystal. This could be found very common in American or European countries where temperature go below freezing temperature, and this is an example of protein crystal which we will bring many times through the courses. First, let us define an ideal crystal. An ideal crystal is the ordered deposition of molecules such that there exists a regular repetition of a pattern in 3D space, where this repetition extends over a distance equal to or greater than 1000 of molecular dimension. That means, when you think about an ideal crystal, you could think about a point, you take a point, you take a pencil, you take a pen and arrange millions of them in several lines and make layers. Then you get a complete thing, a complete structure, the holistic picture, define crystal, and that is a ideal crystal. But the real crystal is not always ideal. The real crystal is less than perfect. The periodicity is not always good. Imperfections are often caused by impurities and the effect of temperature. Higher temperature make perturbance, disturbance, and the ordered nature disturbed. Now, protein crystals like any other crystal of organic, inorganic, small molecules are regular 3D arrays of identical molecules or complexes. One notable difference between small molecule crystal and protein crystal is that very large solvent content of the latter. Protein crystals typically contain 30 to 80% volume by volume solvent. So, you could imagine a crystal of sodium chloride, a crystal of magnesium sulfate, and all they are very composed. But when you are thinking about protein crystal, yes, they are proteins, they are arranging in order, but there are water channels, and as you know, the protein exist and maintain their fold in aqueous solution. So, they maintain that, but they have lots of water channels inside the crystal containing 30 to 80% of the volume occupied by water or solvent. A small fraction of the protein surface is involved in crystal contacts. We will look at them when we go to the unit cell of the crystal. We will see the surfaces. The surface is involved in the crystal contact, the rest being pretty much in solution. As a consequence, protein crystals are very soft, they are fragile, and they are easy to crush. If you have a protein crystal, I am sure you all could imagine diamond. If you are a girl, you are loving the diamond. If you are a guy, you also love the diamond because you want to give it to someone. So, you all know what is diamond is. You imagine a diamond crystal so hard, and now think about protein crystal. You have a protein crystal and you just crash it, you don't even understand. But you have a salt crystal, you crash it, you understand that a force is there and scratching happened. That is the most important difference. Usually, small conformational changes can take place within the crystal lattice without damaging the crystal. So, when you have a crystal of protein, if you add small molecule, if you add other protein, and that goes into and form the crystal, there are small conformational changes observed, but there might be very large structural changes which have seen in many cases by adding of small molecule or protein molecule and mainly small molecules. Therefore, enzyme crystals are very often active as catalyst. Many people, this is very important, many people think when the protein goes inside the crystal, it is a frozen condition because the outcome of the protein crystal is a static structure. But when people have done experiments, for example, I personally have a good experience where we are working with an enzyme beta lactamase, and beta lactamase is an enzyme which have something very interesting. You know, enzymes generally not working or reacting with the substrate directly. This enzyme does. Enzyme beta lactamase interact covalently with substrates beta lactam like penicillin. So, when you have a crystal of beta lactamase, if you add a molecule of beta lactamase inhibitor, you find the beta lactamase react with that compound and covalently linked there. That proves that enzymes are active even when they are in crystal, they are not frozen in the truest sense of the term. This is very interesting to understand biology. Anisotropic physical properties like birefringence due to anisotropic refraction indices. What is birefringence? If you put the crystal inside the microscope and put polarized light, you see that color would be produced. That is called birefringence. So, these are some condition, some results of protein crystallization experiment. You see clear drop, you will many times see clear drop, you see precipitate, you see needle clusters, you see skin like precipitation, quasicrystals, plates, microcrystals, rod cluster and what not. But with all those work starting from, as I told, starting from the gene to protein, a huge journey which I am going to discuss now, and then you do the work with crystallization, at the end you will happy when you will see the single crystal. That is always the dream of a crystallographer, to find a single protein crystal and go further. Whenever we look under the microscope and see a single crystal, we say, Eureka, we got it. X-ray crystallography, as I told, evidence that solution and crystal structures are similar when people have done structure solution using NMR and X-ray, the two techniques. X-ray, as I already told, take protein and make it in frozen condition, whereas NMR deal protein in solution. When people have take two structure, the same molecule, solved by two techniques, they have found that these structures are similar. Here there is an example. This one is a 2JZ2 is a structure solved by NMR technique, and 3C4S is a structure which is solved by protein crystallography technique. They are the same protein, SS10352 protein from Synecystis species, and you see that when you compare, I will show you how to compare these two proteins in the latter part of PDB and visualization. So, you will see that when you align those, you will see similar coordinates in both the structures. Many macromolecules are still functional in the crystalline state. This is what I was talking about. When you are like having condition, you do assay, you will find that proteins are still behaving enzymatically active inside the crystal. That means the protein is still functional. So, there are two type of experiments, co-crystallization and soaking. When you do soaking, if your protein is an enzyme which could act with the substrate, and if there is any color production or something, some assay, you will find that the protein is still active, the enzyme is still active inside the crystal. And more importantly, most protein crystals, as I have told earlier, contain 30 to 80% solvent. So, now we will go inside the process details of protein crystallography. I told about, but the initial phase is to start with DNA. We have to take a DNA and we have to go through the entire process of cloning, overexpression. This is overexpression. You see the bands, and you will find that the bands you are finding here is due to overexpression. This is uninduced and this is induced. So, when you get these fat bands, you know that your protein is overexpressed, and then this is the purified protein. So, you get a purified protein, then you go for crystallization. Then if you get a single crystal, you will go for diffraction, and then when you get the diffraction data, you will process the diffraction data, you will get electron density, and then from the electron density you will build up the model, and the model is your final goal towards getting the structure solved by the technique of X-ray crystallography. So, I will talk about the details, because a lot of people think crystallography means starting from a protein to go for crystal. Behind getting the protein, there is a lot of struggle there, and it is good to know about those stories. But because it is a structural biology course, I will not definitely go into the detailed technique of the molecular biology, but I will definitely represent bacterial cloning as a simplest system, so that you could understand the entire process and the process details. So, one most important part is which gene you want to make protein and go for crystallography. So, finding, optimizing and preparing the target gene. Finding means you have to do background work, you or your collaborators who are involved in that would find out that a protein whose gene they are working is important, is critical. That is why you know that you have to solve the structure to get the atomic resolution to understand the mechanism of how this protein works. So, the first step is to choose or identify the gene. Okay. Then what to do? then you have to go for primer designing. What is primer designing? Because you want to clone, so you want two things. One, you want your gene to have included with restriction enzyme site. Restriction enzymes are enzymes which actually helps to cut at a particular site of DNA, and by that they make a nick in two sides. Here the A chain is elongated, here the B chain is elongated among the DNA. So these staggered cuts are important because they will go and match with the staggered cut of the plasmid vector where you want to clone it. So that's why you need to do the primer designing. Then after designing the primer, you will do PCR. By PCR, your one copy of your gene would be going to 2 copy, 4, 8, 16, 32, 64, 128, 256, 512, 1024. In that way, each cycle. So if you ask for 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 10 cycle, you will get 1024 copy. You do more, you will get 2048, 4096 in that way. So you get a lot of your DNA now by doing the designing of the primer and by performing PCR. And now your DNA fragment have this restriction site involved where a specific restriction enzyme will cut, and the same specific restriction enzyme will make the same specific cut in the vector where you want to clone. So these are common things. More importantly, as a structural biologist, you also do the optimization. What is optimization? Optimization is, suppose you have a protein. How you know before study? There are techniques, computational techniques called homology modeling, ab initio, and gene threading or fold recognition. Mainly the techniques of homology modeling and fold recognition helps you to get a predicted model of your protein. If you know the gene sequence, you know the protein sequence, and now you know the approximate protein structure. If you see some of the portion of your protein are not well structured, you could identify that part, and those parts you could take out from your gene. Why you do that? By doing that, you make the structure more compact, and that will ensure that later when you are going to overexpress the protein, you get more product. When you are going to crystallize the protein, you will get better chance of getting a crystal. Why? I will discuss them on that part. So we talked about finding a gene, optimizing it, and preparing the target gene. The next thing is to get a optimized expression system. When it is bacteria, the handful choice is E. coli. Why? Because E. coli is an unicellular organism, pretty much well known, well studied. There are no ethical concern about growing, manipulating, killing bacterial cell, unlike when you do it with mouse or chimpanzee or higher organisms. They are able to reproduce and grow very rapidly, doubling time its population about every 20 minutes. They say it's 20 minutes, but there is overlapping population. I am not going into the molecular biology, check it. Actually, E. coli double at an average in 9 minutes, though theoretically it is 20 minutes. This is helpful in research to get subsequent generation within a short time. They can survive and adaptive to variable growth conditions. So, as a bacteria they are adaptive, but on the other hand we know that what condition is good for them. So, they are well known. Most of their strains, E. coli, I say E. SRHU as the genus and coli is the species. So, I say about them, but strain could be different. Okay. But even if you get any strain of E. coli, generally they are not harmful. They can be manipulated and engineered easily. So, when we are doing enzyme engineering and all, we could also engineer them through our studies. Mutants, when you are making, are easily obtained using established method and screening techniques, which has enabled many biochemical processes to be linked to the molecular genetic level. So, when you are making changes, when you are doing protein engineering, metabolic engineering, this is the choice. That is why E. coli, when you are working in bacterial system, is the choice of work. Then choice of plasmid as cloning vector, we could see if there is DNA replication origin, it is necessary for the vector and its linked recombinant sequence to replicate inside the host organism, which is E. coli. One or more unique restriction endonuclease recognition site, the restriction enzyme which I was talking about, as sites where foreign DNA may be introduced, because you have to introduce your DNA to this cloning vector. A selectable genetic marker that can be used to enable the survival of cells that have taken up vector sequences. So, when you are growing that E. coli, you have to differentiate that E. coli from other E. coli. So, if you have a gene like beta-lactamase, that would save you from a beta-lactam like penicillin. So, that is the selection. So, if you have a wild type E. coli and you have your E. coli, you grow them in ampicillin, penicillin, the vector containing E. coli will survive, whereas the wild type E. coli would not survive. A tag gene that can be used to screen cell containing the foreign DNA. So, the marker help you to differentiate between wild type and the vector, but then when you have the clone. So, what I mean by clone, you have the vector, you cut the vector, so you make a staggered cut. So, here is the staggered cut and here is the staggered cut. Your gene also have the staggering. So, then when you clone, you have this is your gene inserted. How you select between the wild type E. coli, vector contain E. coli and vector plus your gene. That is why you need a tag gene. So, ORI, endonuclease site and selectable genetic marker, that is what we need. Choice of plasmid as cloning vector, small size, easy to manipulate and isolate, circular, more stable, generally easy to handle, replication independent of host cell, several copies may be present, facilitated replication and frequently have sorry, it is not antibody, it is antibiotic resistance. So, how you do the cloning? It generally involves 5 steps. Enzyme restriction digest to target DNA sample, I talked about this. Enzyme restriction digest to optimize vector, I talked about this too. Ligation of DNA sample products and plasmid vector, you have these, then you ligate them using DNA ligase. Transformation with the ligation product. So, now you have the DNA content vector, you have this DNA content vector, you have to send it inside the cell of E. coli, where E. coli genome is there, you have to put it here. That's called transformation and growth on agar plates with selection of antibiotic resistance. So, this is what I talked about, the growth on agar plate with the selection for antibiotic resistance, which will help ensuring that the vector is there as well the gene is there. So, we generally use two markers to ensure the presence of the vector with one marker, to ensure the presence of the gene with the second marker. Expression vectors, common factor like multiple cloning site, origin, selection marker, which we have already told in the cloning vector, promoter, operator, terminator, they have to be there. Start and stop codons should be there, but in addition, for a expression vector, they should have tightly regulated promoter. That means, see, you are working with one gene product and you want that to be carry out in E. coli, but if you see E. coli have its own 5000 gene product. So, actually with the law of average, your product should come at 1 by 5001 of the total mass of protein produced by E. coli. That could not be economical. So, you have to do something. This something is the induction, where you use generally lac UV promoter to stimulate transcription. The artificial inducer IPTG. IPTG is a stable molecule which mimic galactose. So, now this will be a non-breakable compound. So, once it sit on the lac operon, it constitutively push the operon to produce the protein it is induced with. So, you have already designed it, you have already put your protein there, your protein's gene sequence. So, that would be constitutively produced, and generally you get the 80% mass of the 80% the total mass is your protein. That is what the importance here. Tags for identification and purification, you get 80%, but still there are these 5000 protein and other impurities, you have to purify them. So, you need to have some engineered tag like his tag, GST tag and all to be there, so that you could use that engineering technique to further going through chromatography to purify your protein, because for those techniques, for crystallography, for NMR, for cryo, you need very pure protein. Then solubilization assisting tags, there are some tags like chaperone and all stabilizing protein, which helps your protein to solubilize. Solubilization is very important for optimum production. Now, going to the protein purification, there are chromatography techniques. For size based separation, there is gel filtration chromatography or size inclusion chromatography. For charge based separation, ion exchange chromatography. For specific binding related separation, there is affinity chromatography. Gel filtration chromatography, if you see that it is a column, every chromatography have a column, and you see the beads have, there are several beads with different size, and the size have the key here. So, the column beads have different size pores, and as the column flows, you put the protein in the column, it flows there, large protein excluded from pores and therefore flow rapidly, and small protein enter pores that flow slowly. This is a very funny situation, you know. Entire life you know that if you are overweight, you reach slowly. Who is underweight, they could run faster, and they could reach quicker. Not in size exclusion chromatography. Here they have an amazing condition. What is the condition? So, imagine you are running a competition of 1 kilometer, but instead of free running, there are cylinders. You have to go inside the cylinder and then run through the cylinder, come out, again go to the next cylinder. In that way, or pipes you have to go. Okay. Here also another fun is there. Suppose you are fat enough that you cannot enter inside the pipe, then you are allowed not to go into the pipe. You understand? So, you are running in a competition of 1 kilometer and the condition is there are several pipes, you have to go through the pipes. Okay. But if you are fat enough that you are not, you could not be entering in the pipe, then you are allowed not to go into the pipe. So, in most of such cases, people who are overweight, they finish first. That's happened in size exclusion chromatography or gel filtration chromatography. The large protein excluded from pores and therefore flow rapidly. Small proteins enter pores and flow slowly. Coming to ion exchange chromatography, separation by charge. If you see the charge beads, they are positively charged or negatively charged. So, beads have charge group, anion exchange and cation exchange. Positive charge bind acidic amino acids and negative charge bind basic amino acids. Different protein bind with different affinity depending on their contents of positive or negative charge amino acids. Eluted with increasing amount of salt. Once the protein binds there, then more several charges are given, so they are coming out. Different protein elute at different salt concentration and that is how they are differentiated. Coming to affinity chromatography. Affinity chromatography is one of the most diverse and powerful chromatographic method for purification at specific molecule or a group of molecule from complex mixture. I talked about size exclusion, I talked about anion cation exchange, the ion exchange, but affinity chromatography is special. Why? These two I talked about are like working in general principle. Affinity chromatography, there is no limit. Today you find something which could be attaching to a specific protein, then that would be used as a affinity chromatography column to purify that protein. That is the power of affinity chromatography. It is based on highly specific biological interaction between two molecules, such as interaction between enzyme and substrate, receptor, ligand, antibody, antigen. These interactions, which are typically reversible and are used for purification by placing one of the interacting molecule referred to as affinity ligand into a solid matrix to create a stationary phase while a target molecule is in the mobile phase. Your protein is in the solution, that is the mobile phase, and the column is having agarose with the bead, that is the stationary phase. Many of the commonly used ligand coupled to affinity matrices are now commercially available and are ready to use. I will talk about one which is nickel affinity chromatography. As I told, in time of the genetic engineering, we include the sequence of 6 histidine amino acids, so that these 6 histidine amino acids will come at the end of the protein. Okay. Now, when we pass it through nickel affinity chromatography column, the histidines would be attaching with the nickel. So, the histidine bound protein would be specifically bound to this column, other protein would be flow through. The column contains a compound of nickel that helps to purify the protein. Histidine in the protein forms a physical bond with nickel, thus binding the column. There are 3 stages. First, you have to equilibrate the column, then you have to put the protein which will be binding, that is the binding phase, equilibration phase, binding phase, and then you will use imidazole, imidazole if you see is looking or mimicking the same chemistry like histidine, because histidine have a imidazole ring, but you use higher concentration of imidazole, so your protein would be eluted. That is how affinity chromatography works. So, now the protein is ready for crystallization. What you need, three things is very important. One, high concentration of protein is needed for crystallography. Homogeneous and stable, protein have to be stable and protein have to be homogeneous. You cannot have impurity. If you have impurity or if you have second protein present, your protein have a much less chance of crystallization, and you have considerable amount because you have to make a screening. At least we make 500 to 1000 screen condition. So, you need good amount of protein if you want to go for crystallization. Protein crystallization is the process of formation of a regular array of individual protein molecule stabilized by crystal contacts. If the crystal is sufficiently ordered, it will diffract, then we go for the next step. Some proteins naturally form crystalline arrays, like aquaporine is a protein which present in the lens part of the eye. It actually crystallizes automatically. There is protein like myoglobin, lysozyme who are very crystallizable. Anywhere we are going, we should know about the history. The history of protein crystal growth started about 180 years ago. Think about 180 years so far is recorded. There are other attempts which were not at all successful, so not recorded. The first published observation of the crystallization of the protein appears to be by Hunfield in 1840 of the protein hemoglobin from the earthworm. This observation clearly stated that protein crystal can be produced by the controlled evaporation of a concentrated protein solution. Remind the term controlled evaporation, because that is what we are going to follow when we are going in depth and understanding the principle behind protein crystallization. That is, protein crystal can be produced by slow dehydration. For the next 15 years, most of the crystal obtained from the blood of several animals were found to be by chance. The first person who actually devised successful and reproducible method for growth of hemoglobin crystal was Funke. You could see him, Funke, and very interestingly, you might have a question in mind, why these guys were actually trying hemoglobin? Because that time people don't have protein purification techniques. What they know, that if you get blood, you will get a lot of hemoglobin. Because of its red color, you could understand that the protein is there. That is what his secret behind. The first enzyme was crystallized by James Summer in 1926, followed by which was urease, followed by the crystallization of pepsin in 1930 by John Northrop. And these two guys got Nobel Prize in Chemistry in 1946. For Sumner, it is for the discovery that enzymes can be crystallized. This is a very interesting and revolutionary step towards the development of the technique, which is protein crystallography. And then Northrop for the preparation of enzymes, and more importantly, he have worked on viruses, introduced the virus proteins in pure form. In 1934, John Desmond Bernal and his student Dorothy Hodgkin discovered the protein crystals surrounded by their mother liquors gave better diffraction patterns than dried crystals. So, up to now people did not understand how to continue the process. They found that it is the mother liquor, I am talking about mother liquor further, that would be helpful. using pepsin, they were the first to discern the diffraction pattern of a wet globular protein. So, this is the first attempt where they use X-ray to get a diffraction pattern. They could not solve yet. Prior to Bernal and Hodgkin, protein crystallography had only been performed in dry condition with inconsistent and unreliable results. This is the first X-ray diffraction pattern. It is not the solution yet. It is the first X-ray diffraction pattern of a protein crystal. And the start of the era, the start of protein crystallography in 1958, the structure of myoglobin, a red protein containing heme. Again, I talked about hemoglobin. Myoglobin is also another protein which having iron, so it color red, and that is why it is easy to identify between different proteins. Determined by X-ray crystallography was first reported by John Kendrew. Kendrew shared the 1962 Nobel Prize in Chemistry with Max Peruz for this discovery, and if you see here, this is what Kendrew's model and this is what like Max Peruz model. So, we will talk about this later, but this would be the end of today's class. Thank you for listening, and I would continue from this to the in the next class. Thank you very much.